All right, welcome back and congratulations to everyone who won seeds last week. This week is another giveaway and actually the biggest one yet. We are giving away 35 packs of seeds to get as many people growing this year as possible. This week is also 420, which is like Christmas in the weed world if you didn't know. I thought, what better topic to discuss on 420 week than the entourage effect? This amazing synergy that the cannabis plant produces, including terpenes, cannabinoids, flavorants, flavonoids, all these beautiful compounds that work together to increase the medicinal benefits of this plant, although it can be extremely hard to study in the laboratory. I will warn you that this is a very science-heavy episode. We're going to talk a lot about neurochemistry and how things work in the brain. But hey, this is a science podcast, so you kind of know what you're in for. Other topics that we discuss in this episode, other than the entourage effect, are things like how CBD actually works in the brain. What receptors does it act on? How CBD can help with concussions or preventative medicine, and so much more. As always, if you're a regular listener to this podcast, please consider taking whatever hand you are not holding your joint with and slamming a 5-star review wherever you listen to this podcast. Or if you're watching on YouTube, make sure you subscribe. It helps a ton and literally only takes a second. And I don't know about you, but I am growing a ton of 1-to-1 strains this year to get more CBD into my system because it really is a magical compound and complements the effects of THC so well. So make sure that you take a sip of water, you unclench your jaw, and you smell some terpene-rich bud as you listen to Dr. Josh Kaplan and I for the next hour or so. All right, so welcome back to the Bioactive Podcast. I'm your host, Dr. Riley Kirk, and I'm here today with Dr. Josh Kaplan. So welcome to the show, Dr. Kaplan, and if you wouldn't mind just giving a little more of an introduction on who you are, why you got interested in cannabis research, and what your lab currently studies.
Yeah, well, thank you very much for having me, Riley. I'm excited to be here. So I work at Western Washington University. It's an undergraduate research institution tucked in the northwest corner of the U.S., pretty close to the border of Canada. And we're in a state with legal cannabis, and there's a lot of interest around cannabis, including the potential therapeutic benefits for it. And so that's really what my lab primarily studies. But I didn't get into science or neuroscience to study cannabis. That was never my mission. But when I was working as a postdoctoral fellow back at the University of Washington, now quite a few years ago, I had joined because I was interested in studying a rare form of pediatric epilepsy. This lab was one of their many studies was investigating new therapeutic strategies for targeting this rare form of epilepsy because unfortunately the children who were suffering from it didn't have any available medications that were working for them. So I joined the lab, started doing my own work, and shortly after I got there, they said, Josh, you have a background in behavioral neuroscience. Do you mind just testing this new product that we have been given and see if it works on our mice? We had these genetic models of the pediatric epilepsy that we were studying. So I said, sure, why not? Let's give it a try. It turned out to be remarkably effective. It reduced seizures substantially and also improved a lot of the other behaviors that were comorbid with the disorder. Turned out that drug that they had given us was cannabidiol. At the time, I'd never heard about cannabidiol, didn't know really anything about it. But that completely shifted my research trajectory from that moment on. And so the rest of my time during my postdoctoral fellowship was studying cannabidiol, trying to identify, what is the range of its effects across a variety of disorders, including autism spectrum disorder, and trying to identify the mechanism by which it was working. How is it affecting brain function in a way that reduces seizures, but also can be harnessed to treat a variety of other conditions? And that's the focus that I brought to Western Washington University and what my lab is currently investigating, which is what is CBD doing? in the brain? How is it working? How can it be harnessed to treat numerous different conditions? And what are ways of improving it? And a major focus of our lab is in the optimization arm of it. So combining CBD with other chemicals produced by the cannabis plant, colloquially we call these terpenes, right? To see what combinations will lead to the best medicines. And that's really what our current focus is today.
Awesome. So studying the entourage effect, which I think a lot of people definitely who listen to this podcast are very big believers in. And if I study natural products for my whole life, and the entourage effect is a huge component of many natural products, including cannabis. But I think with cannabis, it's incredibly promising. I would love to go back to CBD and epilepsy for a little bit. This condition you were talking about, we're talking about Dravet syndrome, correct? With seizures happening in children, and it's a very, very serious condition. As you said, there wasn't really any treatment for it, so that's a huge priority in research. Can we kind of talk about the research that you did with mice and how we kind of started to learn more about the mechanism of CBD? CBD is a very promiscuous molecule. It can interact with a lot of different receptors, so really locking down on that pharmacology can be extremely difficult. So how did you go about exploring CBD's pathway and how it could be helpful for Dravet syndrome? And then we can also talk about autism, which I didn't know was linked to Dravet syndrome as one of these, one of these symptoms essentially that is part of it.
Mm-hmm. Well, yeah, there are a number of things to unpack here. And, even just starting with the idea that when you have a genetic mutation that affects a protein in the brain, in the case of Dravet syndrome, it's a particular protein that affects the inhibitory function of the brain, which is a very important element in normal brain function. And when that breaks down, you can get numerous symptoms such as epileptic seizures, or on the similar end of that spectrum, autism spectrum disorder, which results from a similar imbalance in brain function. So, one of the things that I got interested in CBD for was that it wasn't acting on traditional targets that had been investigated by the kind of traditional medications that were out there. And so I felt like its varied pharmacodynamic effects, that is the targets that it's acting upon in the brain, enables it to potentially target a variety of other disorders that are otherwise poorly controlled with the medications that we have. With whether we're talking epilepsy, depression, or a whole host of other conditions, there seem to be about 20% of the folks who suffer from them that are non-responders to the medications that are available. And I'm really interested in finding medicines for that 20%. And for the rest as well, if they're seeking alternative approaches. But that 20% is really what motivates me. And I think CBD represents a potential way of getting at those otherwise non-responders.
Not to interrupt you, sorry, but you mentioned the inhibitory neurotransmitter, that part of the brain. And that can be a little confusing because you have excitatory and inhibitory. And if it's affecting inhibitory, would that mean that the brain is too excited and that's what's leading to these seizures potentially?
Right. You can kind of think of your brain being in this relative balance between excitation and inhibition, almost like a seesaw, right? Where if you have too much excitation, that's problematic because that can contribute to seizures or a host of other disorders. If you have too little excitation, that can also be problematic for depending on where in the brain we're looking. And restoring that balance is important for any therapeutic approach. And so that was the approach that we took when we were looking at this pediatric epilepsy, which you named as Dravet syndrome. That's one of the common types of this pediatric epilepsy that can be treated by CBD, but there are others now as well. And the brain of these children, as well as our genetic mouse model of pediatric epilepsy, displayed this imbalance. One of the ways that you can approach this is you can study patients, right? Patient population who suffers from this disorder. There's a famous case of a young girl named Charlotte Figi out in Colorado who was suffering from Dravet syndrome. She was experiencing hundreds of seizures a week. It was really scary for her family. And while all the medications she had tried were not working, her family kind of threw a Hail Mary and said, what else can we do? And got connected with I believe the Stanley brothers who were producing Charlotte's Web at the time. And they got access to a strain of cannabis known as Hippie's Disappointment, which got the name because it didn't have much in the way of THC. It was rich in CBD, and that seemed to be very effective at reducing Charlotte's seizures, and really kind of paved the way for this empirical investigation that moved to the research labs to identify, does it really work? How does it work? And also coincided with the clinical trials that were going on at the time. So, yeah, I mean, we came at it from a preclinical approach where we looked at the literature and said, okay, well, why are people suffering from this disorder? Well, they have a genetic mutation that affects a very specific part of their DNA, a gene that encodes a protein in their brain that regulates this inhibitory function. And so by making it less effective, now that balance in the brain is tipped, and it's tipped towards excitation in particular brain regions that could promote seizures. So my approach was to look in that brain region, specifically a brain region known as the hippocampus. For those who have taken an introduction to psychology course at some point in their life, they may have heard of the famous story of HM, who had his hippocampi removed and therefore was left with the inability to form long-term memories. The reason he had his hippocampus removed in the first place was because he was suffering from intractable epileptic seizures. And so we turned to this brain region to study CBD's effects. And interestingly, so take a step back, one of the primary research methodologies that I use is a technique known as slice electrophysiology. Recognize that's a long word that basically means I record the communication patterns between the brain cells, which we call neurons, and how they could be affected by drugs such as CBD. So I was using this technique at CBD's effects on the communication patterns between neurons in our mouse model of Dravet syndrome. Interestingly, in the mouse model, or just your standard wild type healthy mouse model, CBD wasn't actually doing all that much, maybe a little bit here or there. But when we exposed the brains from the Dravet syndrome mice, the effect of CBD was substantial. It really increased inhibition, so basically rescued the deficit. And so getting at your question of, well, how did you go about this? It was really just, let's put on CBD, let's see what happens, and then let's start to identify a potential target by which it's working. And so one way to do this is to say, if you believe that, you were saying earlier, CBD, maybe it's acting on the endocannabinoid receptors. Well, you can block the endocannabinoid receptors with another drug and see, does the effect of CBD go away? In our case, it didn't, because it wasn't being mediated by the endocannabinoid receptors. Instead, we use this approach to find that it was seemed to be mediated by targeting a specific type of receptor with a terribly boring name known as the GPR55 receptor.
It is a pretty boring name.
It needs a better PR agent.
Yeah, so it's a G protein coupled receptor 55, which people talk about that as part of the endocannabinoid but it's not necessarily a cannabinoid 1, cannabinoid 2, or cannabinoid, quote, 3 receptor. But it's
Correct.
Within that general, it's related in some way, or at least it's been hypothesized to be related in that system.
Right, and it can be activated by other lipids, which is maybe one of the reasons why it's thought to be in that category of endocannabinoids, which are lipids found within our body. And so we found that CBD was acting upon that receptor to convey its anti-epileptic benefits. At least that's what our data suggest. Cool. But this just represents one of many targets of CBD. there have been over 65, probably at this point now 70 plus targets of CBD that have been identified in the brain and body. Some of them are proteins in the brain, others are enzymes. There's a whole host of different targets. And that's the exciting part to me, because we don't necessarily know which targets are going to be effective for a particular condition, but it gives us a whole new toolbox for investigating disorders that otherwise don't have effective therapies.
Did you analyze how it was interacting with the GPR55 as an agonist, as an antagonist, as somewhere in between? Like, what was that interaction like?
Yeah, our data suggests that it's acting as an antagonist, and this fits with other research, which we're using other models of investigation to show that it was acting as an antagonist as well. So that's our belief.
So, turning off the receptor instead of turning on the receptor, just for simplicity purposes. And how is GPR55 related to this excitability complex with seizures? Is that is that preventing the neuron from getting too excited, blocking something else from binding there? Or do we know the answers to that?
We don't know the exact answer. However, we do know that by blocking this receptor, particularly in this brain region, I can't even speak to how it's working elsewhere in the brain, but within the hippocampus, this region of interest, which seems to serve as an important gate for signaling through the brain, and the breakdown of that gate promotes seizures. Blocking this receptor seems to enhance the brake on that activity. So it increases inhibition and reduces activity through this hippocampal region, which can get out of control and contribute to seizures. So we've seen that CBD prevents just spontaneous seizures from arising, but also can suppress seizures when they've already happened. So as they're occurring, if CBD is in the body, it can reduce the severity and the duration of those seizures. And that's been observed in humans as well, who have gone through numerous clinical trials looking at the efficacy of CBD, not just in Dravet syndrome anymore, but across a variety of different epileptic conditions.
Wow, that is amazing. So it can be used potentially as a preventative medicine. And is this all working through GPR55, or do we think that there's other receptors involved in these different, whether it's helping prevent the severity of a seizure versus, other components that we're talking about here? Is that all through potentially GPR55?
It's a great question, and I wish I had a more thorough answer for you. The electrophysiology experiments suggest that GPR55 is a key player. However, my rational thinking tells us that it's probably more than just that, because why would there only be one target out of those 70 potential targets that are influencing the brain activity here? My guess is it's a whole bunch of them, but this GPR55 seemed to be a key contributor to that. What's interesting, though, and one of the things that has motivated my research and research we're currently conducting in the lab, is the challenge that arose from treating seizures with CBD, which is that in order to get seizure reduction, you need a pretty high dose of CBD. In our animal models, it's pretty darn high, equivalent to what would be on the order of almost grams of CBD per day. And that's, some of these children who are taking CBD to treat their seizures are taking upwards of 500 milligrams or more per day, even with their small body size. However, to treat some of the other comorbid conditions of epilepsy, as well as other conditions that people might suffer from and use CBD to treat, like anxiety, depression, social behaviors like autism spectrum disorder, requires a much lower dose. And actually, you lose some of the therapeutic benefits for those conditions if you escalate to the dose needed to reduce the seizure activity.
Interesting.
So we have this dosing problem that we can't simultaneously treat multiple symptoms of a given condition simultaneously. And so one of the things that motivates me is, harnessing this entourage effect to extend the therapeutic dose range. And I think there's that would really substantially improve the therapeutic efficacy of CBD-based medicines, and make it more reliable for a variety of different people and conditions, but also make it more effective for treating the range of symptoms that one may be suffering from due to even a single genetic mutation.
Oh, yeah. So is this something that your lab's looking at? Either like other compounds in cannabis that act on GPR55, or maybe other receptors like your GABAergic system that might be involved in seizures? Or I know you mentioned during one of your interviews that I was looking at preparing for this interview, talking about also utilizing your own endocannabinoid system and potentially manipulating the levels of the endocannabinoids our body makes, like 2-AG or AEA, and how that could potentially lower the dose needed of CBD?
Mm-hmm. Yeah. It really depends on the symptoms that we're trying to treat. And for instance, one of our targets is the anandamide arm of the endocannabinoid system. One of these abundant endocannabinoids that's produced by our body. It seems to be really important in mental health across a number of different, conditions. Great example is the case study of this Scottish grandmother, Jo Cameron from Scotland, who anecdotally reported very low levels of anxiety, very little pain, even just described childbirth as a tickle. And you hear the story and you think, what is going on? And upon genetic analysis, they found that Jo had a mutation that basically rendered the enzyme that broke down anandamide pretty useless. So essentially, she had very high levels of the endocannabinoid anandamide. And it was thought that this contributed to her low levels of anxiety, high pain threshold, that sort of thing.
Wow,
And then
she was like straight vibing, like high on life, just like, why is everyone upset? Childbirth isn't hard. I have 20 kids. Like, it's all good.
Right. And so if you look at the other end of that coin and say, well, what happens if you have low levels of anandamide? You see that to be directly correlated with conditions like depression, anxiety, heightened pain sensitivity, and even autism spectrum disorder. And so we think that we can use anandamide levels as a measure for the potential therapeutic efficacy of CBD or CBD in combination with different terpenes. And that's one of the platforms that we use. So we recently at our university have been conducting experiments where essentially what we do is we expose our animals to either CBD alone, CBD in combination with different terpenes, and measure anandamide levels in different parts of the brain. It's a really cool new technique that I'm excited about. I was never trained in it before, but it's a mass spectrometry technique. Cool. Where we can essentially take a two-dimensional image of the brain and say, where is this particular molecule being expressed? So we can look at anandamide, we can look at 2-AG, and use that as a metric and say, is this combination boosting levels of this therapeutic compound? And so that's one of our primary screening tools we use to discover better medicine.
Wow. Quick question. Is this MALDI-TOF we're talking about, or a different?
It is.
Okay. That is so cool. That is so just a type of mass spec. We won't get into detail about that, but I'm a little bit of a mass spec nerd. So this is really interesting. Is this kind of a potential sign that we could be moving towards more of like personalized medicine with, each person having different endocannabinoid tone and potentially being able to be dosed differently, or finding products that work best for your brain instead of everybody taking the same dose and the same products and hoping for the same effect or output?
I mean, it is a step in the right direction. The dosing questions are so challenging.
Yeah.
Because CBD seems to have a pretty narrow inverted U dose response relationship in which low doses may not have much of an effect. High doses, also you lose that effect. There's some sort of Goldilocks level of CBD that treats the particular condition that you're interested in. For instance, we have observed that very low doses of CBD are effective at improving the social deficits in autism spectrum disorder. Higher amounts of CBD are needed to reduce generalized anxiety, and then even higher levels of CBD are needed to treat seizures. But with each escalating dose, you might lose the efficacy of those symptoms at lower doses. So it creates a challenge, and knowing what your Goldilocks range is a very challenging task. And it really, at this point, for people, requires a little bit of trial and error, unfortunately.
Yeah, and we've seen that a lot. We've talked to other people on this podcast that are talking about cannabis and anxiety, and in talking about needing maybe like 200 milligrams, but not obviously, but also a part of this is taking CBD in isolation versus taking a full spectrum product versus taking a broad spectrum product. And you're so right. I think the answer always comes back to this trial and error, which, you know, if you do have seizures, if you do have really tough anxiety, like that might not be something that you're comfortable with being like, oh man, I gotta do a little experiment on myself for the next few weeks to see if anything works. So I do hope that research like your lab is going to start to, you know, hone in on that in the future, or at least develop some sort of like protocol so people at least feel prepared to make those decisions themselves, or physicians, if physicians are interested.
So we did a couple studies just looking very basically at this, where we looked at what we call sub-threshold levels. So basically the amount of either CBD or a given terpene that on its own doesn't have any effect. It's below what we would predict to have a therapeutic benefit. And then when we put them together, we saw the benefit. So I do, you know, to me, that suggests that adding the terpenes into it, going closer towards a more whole plant pharmacological approach, at least increases the probability that someone would get the benefit they're seeking, should that benefit be real and exist. So yeah, I wish I had clear guidelines for here's how much to take at what time, but it differs by the individual. It differs by the sex of the individual and hormonal levels. It differs by the way in which you consume it, whether it's edible, sublingual, inhalation. You know, there's so many factors with cannabis, in part because the markets have established a wide range of consumption methods and products, which I think is great, but it also makes it hard to give clear guidelines for use.
There's so many variables, especially compared to like the traditional pharmaceutical model of medicine, which is like, you know, you got one thing. So when you are testing terpenes, are you just question on how do you go about this? Are you typically, you know, starting with CBD and then adding one terpene and seeing what happens, adding another one? And are you seeing like significant differences in terpene profiles with some of these outcomes that you're looking at?
Yeah, good question, because we've taken both approaches.
Okay.
We have been collaborating with a company known as Abstracts Tech, and they
love them.
They have synthesized and extracted different terpene profiles from common cannabis strains, your OG Kushes, your Blue Dream, Do-Si-Do, and we've tested those. Yet those terpene blends are essentially comprised of many different terpenes. And we've tested those on their own in addition to CBD, and we have seen benefits of the terpenes themselves, but also the combination with CBD there. We've also looked at individual terpenes. For instance, we just published a study looking at the anxiolytic effects of linalool and limonene, two common cannabis terpenes, both on their own and in combination with CBD. And this gets back to that point I made earlier, that when we looked at it at a sub-threshold level, so below the effect, below a dose of that terpene or CBD that was effective on its own, the combination led to a more robust and reliable effect. So our evidence suggests that the combination will at least, again, increase the likelihood that someone would get a benefit should that benefit exist. And you ask, like, how do we do it? You know, we have different administration methods. So we've moved away from the injection method in mice, in part because I don't know many humans that are injecting these drugs into themselves, and we want to mimic the human use pattern. So
love that. I don't know anyone who's injecting cannabis at this point, but I'm sure there's somebody out there who's tried.
To each their own. But so we have vaporization chambers. We call them our little hot boxes, or at least that's what our students call them.
I love that.
They're often the main stop on the lab tour. So the animals can breathe it in just like a human would. Or we have an edible method. We basically mask the flavor and smell of the terpenes in a bacon flavored dough. And
awesome.
The animals love it. They go nuts for it. They kind of even jump around sometimes when they're about to get it. So they eat it that way. And that's another method that we can control and measure how much they're taking in and the effects that they're having.
Wow. I love that you're using vaporization as the technique, because I see IP, or like the injection, as the route for so many different publications. And as you said, it's like, that's not very relevant. So that's really cool. Working with Abstracts Tech, are you also looking at these other non-terpene compounds, these flavorants that they're looking at that are in the plant? And I guess second question on that, are you also evaluating any component of the entourage effect with other cannabinoids, whether that's CBD and CBG, CBD and CBN, or whatever that combination might be?
Those are important things to look at. Absolutely. The additional phytocannabinoids. We ventured down the CBG world briefly several years ago, but realized we had our hands full with one, with just CBD. I would love to have more time and resources to do it, but at this point we're sticking to CBD. And similarly with abstracts, we have a number of different volatile organic compounds. They're VOCs that they have extracted and produced. And so we are testing several of them, but actually just a couple of weeks ago, we got a nice NIH grant from the National Institute of Health to basically study these entourage effects. And
wow,
so
congratulations. That is huge, massive. I hope you actually get the money because of what's happening in the government, but either way, congratulations.
Thank you. Yeah, the last few days have been a little bit of turmoil, but
oh my gosh, I can't imagine.
I think we're okay at the moment. And so we're really excited about really diving into studying these entourage effects. And we use 3 different main approaches for screening these, because it is a major task. There are so many different terpenes that we can study, so many different phytocannabinoids, and so we can do this with electrophysiology and just measure their impacts on cell signaling. We can do it with the MALDI-TOF mass spectrometry, which you mentioned, where we're looking at like anandamide expression in a 2-dimensional brain, and we also have a really neat collaboration with a biology professor here who's really an immunologist, but one of the things we're interested in is neuroinflammation and the effect of inflammation on the brain and the hopeful therapeutic effects that the cannabinoids can have against the inflammation in the brain. Because after all, inflammation is the seat of many problems that arise, whether it's acute disorders like autism, epilepsy, depression, or neurodegenerative diseases like Alzheimer's. And so we think that this could be a potentially powerful protective strategy as well. And so we've teamed up with this immunologist. Her name is Dr. Annalise Snyder in our biology department. And essentially what we can do is, if the brain is expressing signals of inflammation, there are certain proteins that get expressed that we can actually tag with antibodies, run it through a filter essentially, and say, okay, what type of cells are being expressed, which are active? How does this relate to neuroinflammation, so brain inflammatory states, and what effect are the cannabinoids having on this inflammatory response? And so this serves as another platform for screening the cannabinoid terpene interactions on brain inflammation, which I think is going to be a very valuable future direction for us here.
Whoa, time for the seed giveaway. This week we're giving away 35 packs of seeds from Royal Queen Seeds and Last Green Valley Seeds. So it's the biggest giveaway yet. This giveaway is a little different because it's open to anyone on Instagram, YouTube, or Patreon. And all you need to do to enter is to fill out the Google form that is linked in the show notes of this episode. So there's no magic word, there's no magic phrase, and it's not limited to anyone. So anyone who listens to this podcast and finds that link can enter to win. Just so you know, 22 of these seeds are feminized seeds and 13 of these seeds are photoperiod seeds. And make sure you follow Last Green Valley Seeds and Royal Queen Seeds on Instagram to give them a little love. So thank you for listening and good luck. 100,000%. That's something I studied in graduate school, and it is so needed. It's linked to honestly almost every chronic condition. Neuroinflammation is, and I just need to give you extra props for studying two of the hardest things, which is preventative medicine and the entourage effect, because there are so many challenges with both of those things, and it sounds like your lab's doing a great job doing the research and doing it in a great, like, orthogonal way, doing it in multiple different ways to try to get a better grasp of what's going on. Because if you just used one tool, you might not really be understanding what's going on, but really utilizing everything you have and collaborating is, I think, the way to move forward with that. And I'm sure your students absolutely love doing this type of research too, working with cannabis. Are you able to work with THC at all, or is that limited through the university?
We had a license to do it. It's just one of the things that I have been motivated by throughout my career working with cannabis is extending access to safe and effective therapies for children in particular, or adolescents. And I believe the safest way to do that is to avoid THC because of the potential effects that it can have on the developing brain. Now, sometimes those effects might be selected over the alternative, which is the other medications they might be taking, or the impact that the disease itself could have on the brain.
Yeah.
So some people or some parents may choose to go the THC route for that reason, because they see that as the better alternative to whatever else there is. But I still believe, based on the work that we've done and the literature, that we can achieve many of these therapeutic goals without THC, and that's what's been motivating our work in the CBD space.
Oh, I love that. And CBD has been proven to be so incredibly safe, even at the doses that you were talking about, 500 milligrams a day. I mean, of course, if we can lower that, it's going to be even safer. But I think one of the most remarkable things about CBD is how effective it is and how safe it is. So I think that's really amazing research.
No, I was just going to add to that, if we are thinking about something as a long-term treatment, then you do want it to be safe acutely. One of the I remember reading a study back when I was in college, which I think it was published in the early 90s, which said that people who had taken a bunch of NSAIDs like ibuprofen or Advil had a lower risk of developing neurodegenerative disease like Alzheimer's disease. And they came out and said, look, this is an association or a correlation, but don't go out and just take unlimited amounts of ibuprofen, because that has harmful effects on the body. And that kind of raised my awareness that, like, maybe there is some sort of preventative treatment that's out there, something that maybe reduces inflammation, that could be taken more long-term safely, that, could have these benefits over time. And so, again, that's one of the reasons why I think CBD has potential as a preventative treatment. People are looking at it now for everything from preventing the damage caused by concussion, like in the NFL, or other high-impact sports or high-risk situations, even in the military, or the potential treatment or potential protective effects against neurodegenerative disease. So, yeah, the safety element is really important to me, that it could be used for a long term as opposed to just acute medicine, even though there's value in that. I'm also interested in the long-term potential that it could have.
Oh, I love that. And I love that you just mentioned TBIs and sports and veterans. This is an area that we obviously need more research in, and we have some really exciting clinical trials coming up. And you were a co-author on this amazing review paper that was reviewing TBIs and CBD and potential mechanisms. Would you mind diving into that a little bit? And are there overlapping mechanisms between how CBD might protect the brain with TBI and how it's working with the brain with seizures and autism? Like, are those different? Are they the same? What kind of findings did you bring together in that review?
Yeah. Thanks for bringing up that paper. I found it to be a really interesting deep dive into this literature. We collaborated with a local neurosurgeon who really shared his clinical insight, something that I'm not super familiar with. But, with traumatic brain injury, it's considered the silent epidemic because so many people suffer from traumatic brain injuries and yet maybe don't seek care or help. And so it really goes often unrecognized, but yet can still have really profound long-term consequences because of the what we call the secondary injury with brain injury. You think about the primary injury, that's the actual blunt force that occurs when you bang your head, whether it's an accident or, you get hit hard in football or punched in, whatever that might be, that causes an initial, shearing of neurons. It creates this physical damage in the brain. But what that does is it initiates this cascade of events, which we call the secondary injury. And actually, the secondary injury, while it's there to keep you alive, can also wreak havoc long term. And it can lead to everything from seizures themselves to long-term depression and other increased risk for neurodegenerative disease like early onset Alzheimer's, that sort of thing. So we thought that maybe, CBD as an anti-inflammatory could protect against this damage caused by secondary injuries. So we did a real deep dive, and there's been quite a bit of research into the effects of CBD from simulated traumatic brain injuries in animal models. I'll note here that the actual research in human populations is quite minimal at this point. But there are some promising upcoming clinical trials in at-risk populations, including even athletes who are experiencing chronic brain hits within the National Football League. So there's been a lot of resources pouring into this field. So I'm excited about where it's going. But, if you're suffering from a traumatic brain injury, there are immediate clinical considerations that need to be taken account, often that relate to the brain edema, the swelling that occurs in the brain that compresses the brain and causes further damage itself. So one of the first things is, can CBD reduce that swelling, that inflammation that triggers further damage? And again, coming from the preclinical realm, using animal models of injury, CBD can have effects. It can reduce the number of reactive oxygen species, which have a feed-forward effect on neuroinflammation. It can restore, under some experimental conditions, the blood-brain barrier, which, when that gets ruptured, contributes to further inflammation in the brain.
Because more things can travel into your brain that normally wouldn't travel into your brain and cause that inflammation.
Right. Yeah, the blood-brain barrier, turns out, is really critical in brain health. And things that break down the blood-brain barrier can increase risk for a number of different long-term medical conditions. There is a really neat review paper that came out, now it might be about 8 years ago, which showed that the breakdown of the blood-brain barrier precedes a lot of the other types of morphological symptoms of disorders like Parkinson's disease and Alzheimer's disease. And so you start to wonder, is there a link there? And certainly there seems to be one with the damage caused by traumatic brain injury. So as a potential restorative tool, there has been some studies linking CBD to restoring the blood-brain barrier, restoring cerebral blood flow. And then you start to think about, okay, well, what effect is that inflammation having on the signaling within the brain itself, this excitatory inhibitory balance? And then coming back to that, epilepsy realm that we talked about, by boosting the inhibitory signaling, now you might start to see benefits long term because it's preventing the excessive amount of excitation that could either be contributing to seizures or further damage that can occur. So there's a number of different avenues in which CBD might provide therapeutic effects in the case of traumatic brain injury. All of these are worth noting in humans, and we're studying in humans who suffer from traumatic brain injury. And I am excited that some of the larger organizations of at-risk individuals are starting to consider this as well, because that could lead to the clinical trials needed to really identify the utility. I'd say one major question that people should consider when thinking about this is, does CBD need to be on board in order to get the benefits? So does it need to be in your system before their injury? Ooh. Or if you're injured, is there like a critical time window in which administration of CBD still has protection? Like, are you too late at that point?
And could there be like an inhaler? Like if somebody like immediately gets hit in a football game or something, could you have an administration method that's like more rapid than, or maybe, vaporization or something, or just have a protocol so it happens immediately after, instead of, them having to visit 4 physicians before somebody suggests it. Can you talk about any of these NFL studies, or what's going on, when it was announced, and if they've announced what they plan on doing with these studies?
Yeah. I love that inhaler idea. I don't know. I honestly don't know what the time frame is. Presumably, if it's on board, it could get working right away. And so I don't think, I think that would be better, but not everyone is willing to have CBD in their system all the time. It's maybe more costly. There's not like a major impact like THC on intoxication, or there's no impact. So, but I could still see some of these organizations being like, we don't want people to be using a cannabis product before they go into combat.
I could see that too, unless it was like highly, highly regulated, where it was, the same product, everyone's tested, everyone's taking the same dose. So there was no, wiggle room for potentially taking other types of products. I think they're too strict in general with all cannabis products in sports, but that's just me.
Sure. Yeah, I mean, I would second that as well. But yeah, the NFL studies, so I think it was 2022, they allotted about $1 million to studying CBD. If I recall correctly, kind of where that landed, they were going to study it in veteran athletes and look at kind of the outcome of, how people are responding following a career playing football. But also there is a separate study that would investigate current athletes. And tucked into that was the prevention of TBI, which to me suggests that it would be either an onboard treatment or something they would give in the moment to active players. So I haven't followed up too closely with where they're at and what has been approved. I don't believe they have started, but I believe the protocols have been written and they have allotted money. I believe a year later, they allotted an additional $500,000 to studying. And I apologize if I'm getting this wrong, but to just further investigation within the NFL. The NHL has also been investigating CBD in their veteran athletes. So not active players, but in veterans, because a lot of these individuals are suffering from the long-term consequences of the repeated brain hits. And there's an increased rate of dementias, neurodegenerative disease, high rates of depression, and as a result, substance abuse. I think some of these players are suffering from it. There have been several high-profile cases of athletes who have unfortunately committed suicide, but doing so by shooting themselves in the chest so that their brain can be preserved for research. And, that type of foresight, I mean, as tragic as it is really interesting because they recognize something's wrong.
It's a very, very, very powerful statement. Yeah, that's like, I have chills. That's insane. But it's incredibly sad. And there's also parallels to the military veterans too, and like how we need more research in that population of people, especially if they've experienced severe trauma and TBIs. And, why are we not providing more research with cannabis to these incredibly sensitive populations who have like no support, especially when they leave, these professional sports organizations or the military? It's kind of like they're kind of just left. So I love to see that professional sports are starting to do that. And I hope we see that our government starts to also put money towards military veterans.
Well, and it's been promising that the National Institute of Health is devoting more financial resources towards this type of research.
Awesome.
It just keeps going up year after year into the therapeutic arm of cannabis. For so long, the only way to get money to study cannabis was to study the harm caused by it. So people were writing grants just to study the negative impacts, but we're starting to see that turn to the positive impacts, and I find that promising.
Very promising. And labs like yourself getting NIH grants, that's a huge movement forward in showing that, people do care about the entourage effect. Most consumers are looking for these full spectrum products, and likely for a reason, and following those trends in the industry and really putting them towards preclinical, but also clinical trials eventually, so we can start to put more answers towards us, even if we don't understand like full mechanism of how every single terpene and every single cannabinoid works in the body. We could still measure general outcomes and how they're bettering the quality of life and traumatic brain injury and neuroinflammation and these other important endpoints.
Yeah, thanks.
Yeah, that's awesome. I know you mentioned it a little bit, but we're kind of getting towards the end, and if you have any insight on the challenges that you've experienced studying chemical synergies, studying the cannabis plant, and just obstacles in general, for any other researchers out there, I would love to hear your opinion on that.
Yeah, there are many, and it goes beyond just like strict regulation. I think both the obstacle as well as the excitement around studying this is the vast variety of things to study. Which cannabinoid to choose, which terpene to choose. There's just a laundry list of each of these. And if you are going to study a whole plant, how do you identify what is contributing to the benefits that are being observed? It's easy to think that, oh, well, if this whole plant extract is just rich in CBD or rich in the terpene linalool, that it must be from those elements, but it could be something totally minor. So it really becomes difficult to ascertain which are the contributing factors at each level of analysis. So kind of taking a more systematic approach, we've been learning from the whole plant, but also trying to take a systematic approach in parallel to figure out what is really contributing here. The other piece is the dose challenges. I find that to be, in our lab, really tough, and also interpretationally for people. I mean, I've seen so many articles out there, whether it's in the New York Times or in a peer-reviewed research article that says, is CBD bullshit?
I've seen them too.
Yeah, and you know what? From an outside perspective, I don't blame that analysis, because you look at some of this stuff and you say, it's not working for them. And therefore, it's overblown. And sure, some of the comments and some of the claims are absolutely overblown out there. It is not a cure-all for everything. Yet, I think that we do ourselves a disservice by saying that just because it didn't work in one study at one particular dose doesn't mean that it wouldn't work at a different dose or for a different cohort of individuals with different symptoms or different genetic backgrounds. As I mentioned earlier in the talk, when I was studying CBD initially using this electrophysiology approach, in the healthy wild-type mice, CBD didn't really do all that much. But in the diseased animals, it really did. And we've been observing that in our lab here, that when we impact the brain in some way, whether it's they're stressed out, they're anxious, they have some genetic symptom, genetically caused symptom, that's when we see CBD having big benefits, not under just normal healthy conditions. So that's a challenge, right? Is like, what dose should we be using? When do we acknowledge that this is a dead end because it's not going to work? Or when do we keep pressing to try and see if something actually is there, so that we don't miss it? I think that has been a challenge for us, is trying to choose the roads to go down based on what works and what doesn't, because we just don't know if we have simply missed the dose range or missed the symptoms that we're trying to capture.
And animal models too. It sounds like you had an amazing animal model for the seizure research, but I'm sure some other people doing research might not have as well of a good representative animal model, and therefore maybe not getting, the results that they're, quote, looking for. And just out of curiosity, CBD is a substance just taken by millions of people, right? If not, hundreds of thousands. Is there research that you know of that's trying to leverage what people are already doing and trying to see how well that's working for people and kind of a human model, maybe even more survey-based model, rather than animal model, and trying to kind of pair those together?
Yeah, there are certainly self-report studies where they're generating self-report data. Those are remarkably positive, right? It's like it works in like 95% of the people. Some of that is expectation based, and that's why we try to do the blinded studies where people don't know what they're taking, or we're doing it in animal models to truly identify efficacy. There's whether it's placebo in some cases or not, like as long as it's working, like by all means, go for it.
Agreed. Yeah.
What I don't want is people to spend a whole bunch of money though on and time and wasting other potential resources on something that actually doesn't work for them long term, and they're going to be left out of luck, shortly down the road. So yes, those are informative, and they're certainly informative for our research purposes because I want to study human use patterns and why people are using it. That's one of the neat parts about cannabis is instead of like the traditional medical or drug development approach, which is let's figure this thing out in a lab, translate that up to humans, and then use it in humans, we're actually going the opposite direction, which is what are humans doing? And then let's work backwards. So those are informative from that standpoint, but whether I would hang my hat on that at the end of the day is like, this is the true measure of efficacy, I think those studies are a bit limited in that way.
Would totally agree. Okay. Can I ask you a few questions from the bioactive community on Patreon real quick?
Absolutely.
I believe there's 2 anonymous and 2 non-anonymous. So I'm going to actually start with the anonymous questions. The first one asks, I've seen research that shows that CBD may take a while in order to help you. Is this something that you see in your studies, that people or the animal needs to take it for a given amount of time before it's seen the positive effects?
That's a great question. And it gets into that optimal dose range element that we talked about earlier. We have seen effects in our animal models right away. And in part, I think that's because we tend to use higher doses than what a human might initially use. Just because we're in better control of it. But if you like go to the store and you buy some CBD gummies or something, often the amount in those gummies is quite low, especially if you eat it, and most of that gets broken down by the stomach anyway, and the liver, that you're going to lose a lot of that CBD right off the bat. And so the dose is lower, the concentration is lower in the body. But because CBD has a pretty long half-life, if you start to use it regularly, like on a daily basis, it will add up over time and get into the system to the point that now you're in that optimal dose range. So yeah, and for a lot of human use indications, it does take a while because it takes time to get to that range.
That's a great answer.
Yeah. Okay.
Yeah. Awesome. Okay, second anonymous question. What does an example of a CBD dosing schedule look like? Should people be taking tolerance breaks from CBD? Any insight into CBD and sensory sensitivities in the context of neurodivergence or potentially autism?
The tolerance question is certainly an interesting one. Clearly, that's observed with THC.
Yes.
Hence why we have such escalating potencies anyway. When people are using THC for treating pain, you see tolerance occurring as early as 36 hours after initial use. With CBD, that is still more of an open-ended question. Yet there was one study that sought to address this in the epilepsy field, because after all, that's where a lot of the clinical trials have been spent in the epilepsy field, is in people suffering from epilepsy. And so plus those children as well as adults with epilepsy are taking much higher doses than what would otherwise be used for other conditions. And therefore, the risk of developing tolerance is higher.
Yeah.
In the one study that I can think of, after about 7 months, about 25% of the participants developed tolerance. So that's actually pretty low, especially if we're thinking about THC tolerance developing after merely 36 hours. Here we're talking about 7 months, and it's like barely in a quarter of the participants who are taking upwards of 500 to over a gram of CBD a day. So I think the risk is lower with CBD.
Yeah, that's interesting. I wonder if that has to do with its promiscuous nature on a bunch of different receptors and like not building tolerance as quick. But either way, that's really, I have not read that paper, but that's super, super interesting. Any insights on hypersensitivity with something like autism? I know that you had published in the past with like anxiety and social interactions, but any other hypersensitivities that you want to discuss?
We haven't come across hypersensitivity, but if heightened sensitivity is underlying symptoms of autism, which this kind of intense world hypothesis that might explain some the symptoms in some individuals who have autism spectrum disorder, by dampening that down because it's increasing inhibitory function in the brain, that could actually reduce some of those sensitivities and allow people to interact a little more easily. I actually had a student a couple years ago now who had autism and said, you know what, I read your paper, I tried it for myself, and the only reason I'm here in class is because I've been taking CBD. And I thought that was one of my wow, this might actually be an impactful moment that made me so grateful to be in this field and being able to do this type of work. So for that one individual in that one case study, it was supportive. So I found that to be really neat.
That's incredible. You're definitely changing lives with your research, and I'm sure that's like incredible to see and feel in in person. And like also I'm sure it's a very passionate student of yours if they were reading your papers ahead of time and implementing your research.
I try not to assign them too much in class, but it has come up every now and then. To make a point, of course.
The next question is from James Berard Moore, or JT, and they ask, what functional networks are modulated by CBD and THC, and how does it change with the entourage effect? That's a pretty massive question. I know we've kind of touched on it a little bit, so if you want to just review some of the main things that are occurring in the brain.
Yeah, what functional networks? A lot of them, basically. Yeah. If you're talking about a system that is found all over the brain, whether we're talking about the endocannabinoid system or just the many different targets that these phytocannabinoids act upon, we're talking about brain-wide interactions here. I mean, the endocannabinoid system is one of the most diffusely expressed regions throughout the body, let alone just the brain. So the actions are plenty, and they're found all over. And they can affect everything from systems impacting working memory, reward detection and identification, anxiety and threat detection. So if we're talking THC, I mean, it's going to be a little bit different than CBD. And in some cases, they have opposite effects. But that's worth like an entire hour of this podcast in itself to really dissociate those different components.
Yeah.
So sorry to not get into too much detail.
No, I think you also did get into a lot of detail. I think a lot of this question was answered throughout the podcast. So I think that's a great answer. Okay. Last question is from John H. And they ask, are the varieties of CBD and the entourage effect the same for everyone? Or would my fake example here, experience with CBG and THC, be different than Sally's experience? Is there a list, table, or resources that shows what the entourage effect could be?
I wish we could put together a table or a list, and maybe that's what I'll do next year when I have a little bit more time. No, there isn't a clear list or of all the factors that could influence the effect that these can have. I mean, one thing that we're also studying in our lab is the influence of estrogen levels on the effects that the cannabinoids are having in the different behaviors and things that we are measuring, because it is believed that estrogen can impact the endocannabinoid system in powerful ways. For instance, estrogen has been shown to influence how many of those CB1, those cannabinoid receptors, are expressed and can be activated by either the endocannabinoids or THC itself. So we're doing a very controlled experiment looking at the effects of the cannabinoids at different stages of, for instance, the estrous cycle, and trying to correlate those factors. But that might be very symptom dependent and kind of depending, what the type of medicine somebody is taking could influence that. So we do believe that estrogen levels have an influence. What that means and how the different cannabinoids are interacting with that part remains unknown. Other factors, goodness, I mean, I'm sure there are a whole host of genetic factors that have yet to be revealed, but that's another challenge in the cannabis space, is the amount of variance in the response that people might have to the cannabinoids, because it isn't, they're not just acting on one target, whereas many medications that people might take are specifically activating a target, a GLP-1 agonist, right? As we hear much about in today's news, or a serotonin agonist. But here we're talking about something acting on many different systems, and each one of those systems has its own degree of variance. So the expectation that a combination of CBD and THC, or CBD and linalool, is going to have the same impact on me as it is on you, it's probably misguided, and we have to accept a degree of variance within that outcome. So I wish we had a clear guide for what to expect. We're just not there yet.
Yeah, and I love that you talked about in this episode that example, the clinical example of the woman that was producing too many endocannabinoids. And, we've talked about on this podcast, clinical endocannabinoid deficiency, and then maybe that's called clinical endocannabinoid efficiency. I don't know what that would be called. But I mean, there's clearly this whole spectrum on, our body producing these different levels of receptors and endocannabinoids. And, I'm hoping in the future we'll have the ability to potentially test for that, especially in sensitive populations, and, try to make a more personalized approach towards cannabis. But as you've been reiterating, we're not there yet. We don't really have a resource yet to really describe the entourage effect. But I do think that your research, especially with this new NIH grant, is going to help push that research forward so much. So I'm really excited to see what comes out of that research in the future.
Well, thanks, Riley. I appreciate that.
Yeah. So last things, how can people stay up to date with your research? Do you recommend following your lab website, Twitter? What are the best ways to stay up to date with what you're doing?
Yeah, our lab's website will post kind of the latest news of what we're up to. It deserves a little bit of an update. But yeah, I mean, I do hope to kind of share the exciting stuff that we're doing through that mechanism. We'll also post our most recent publications. That would be the best way to kind of follow up with what we're currently engaging in here at Western Washington.
Awesome. And I'll put that in the show notes of this episode. And the very last thing is, do you have any advice to any future cannabis scientists who want to do what you do, or want to work in a lab like the lab that you do this amazing research in? Any advice for the future generation?
Yeah, just follow what interests you. It makes this game so much more fun to play. Get excited, be interested about the potential, and yeah, just keep hammering away.
I love that advice. Yeah, passionate scientists are the best scientists. So thank you so much for taking the time to be on this podcast, explain your research, explain all of these incredibly complex systems in the brain. I think this episode will be a great resource for people interested in neuropharmacology and all of those interesting aspects.
Cool.
Awesome.
Thanks, Riley.
Absolutely. So mad love, everyone. We'll see you in a couple weeks.